And so this legend goes, for 100 years, in that part of the world, potatoes weren't allowed to be eaten because they made the royal family sick. How cool, if that's true. The second one is about the bringing of potatoes. We already talked about that to the New World. And so I just thought that was cool. From Peru all the way around and back over. So there you go. And that's the person that in Google is attributed with that. And the third thing that really I thought was so neat. So Marie Antoinette, you've already heard, she did the thing in the carriage. People were looking at their hair and they said, "What's that?" It's a potato blossom. And that brought the potato into favor. Wow. Just to think about how we get to eat the things that we do. Wild potatoes of the Andes. I thought this was such a cool picture to realize today's potato. We talked about genetic improvement and how the potato has changed since those wild days. Through that process of agriculture and domestication, I wanted to point out to you, these are the types of potatoes, not only the whites, but now the yellows, the pink fleshed, and the purple fleshed, being developed in many centers, including the center down in San Luis Valley, the Holdings Group, developing these particularly for hopefully their health benefits. So, you know, I'm a nutritionist, and what's a nutritionist say? Oh, potatoes, watch out, eat the other stuff. And then I tell you that it's America's favorite food. That's true. That's what we eat. So I think part of the reasons for discussions like this is use the staple crops. Hey, if people like it, let's empower it to deliver health benefit to the population. And so what are the efforts just to kind of get you to think about the potato in a new light? Opportunities going on in Colorado and other places actually around the world. Change the carbohydrate content of the potato, the type of carbohydrate, change its availability so it could actually be Atkins friendly. People are working on that. Secondly, that phytochemical richness. Now Steve, I'm sorry, but there's another culture out there, maybe, so we know that the purple stuff in grapes probably pretty good. Those purple potatoes are kind of mimicking grape. And Pat Byrne here, he said he worked on that purple corn. Mimics grape. So maybe we won't get that as the fluid variety of our nutrition. But people are working on that and trying to deliver in the potato important health protecting chemicals. So my prediction for you, and this is where I'll stop, and boy am I done well on time. I think in the future, you're going to see, and I'm serious about this, you're going to see the marketing of potato, and I can tell you about potato varieties we're currently using. They actually help our fat mice lose weight. So they'll be marketed for their satiety benefits, awesome, for their anti-inflammatory, anti-cancer, and anti-diabetic activities. The point being, when we put our mind to it, and we in the biomedical sciences work together with people in agriculture, great things can happen. That's it. And now I'm going to reset the timer. And Chris Waters is our next speaker. See, she got her degree from Cornell. She's a plant physiologist, and I don't know if she has a lot of contact with students, so she probably isn't a stress physiologist. Ha ha ha, okay. But she works over at the National Center for Genetic Resources Preservation, I think. Is that correct? That's the title. How about that? So I'm going to set the timer for her. Oops, I gave her a couple extra minutes.
I didn't think I'd need that.
And you probably won't need it. There you go.
Hi. So how many of you have collections, big collections? How many of you have collections? Collections of stamps, coins, cards? We all like to make collections. And collections are pretty interesting in the context of this particular seminar, because collections are, well, really they're a study of diversity. Things are really the same, but they're also really very different. And it's by understanding what's the same and what's different, that's a really basic thing in human understanding. We learn that when we're little children. Some of these things are not like the others. In the final talk, I'm not going to talk about individual crops. What I'd like to do is just change gears a little bit and talk about collections. And it happens that Fort Collins is very famous for, there is a very famous collection right here in Fort Collins. And that's where I work. It's the National Center for Genetic Resources Preservation. It's hard to say. We used to be the National Seed Storage Lab. That's what I say on my phone. But here we are. You can just Google it, NCGRP, and find out all about us. But we are a collection of genetic resources, the basis for what everyone has talked about today. It's a research collection, and so all those wonderful things that you've heard about today in terms of research, most of that germplasm is right here with us. And what we can do with collections is study diversity. You really haven't seen, you've seen slides like this, but not quite presented in this way. When we have collections, we can grow them all up together. This is a common garden experiment. And we can look at the diversity in a removed habitat. This is a picture of lettuce, and you can see, you can see that there's differences in growth type, there's differences in flavor, there's differences in when they flower, there's differences in the seed. Some lettuce is not used for leaf, they're used for oils. There's differences in just about any kind of diversity that you'd want to know about. And that's what we have right here in Fort Collins. We don't usually grow it up like this, but we have it right here and safe and available for research. Really what we are in Fort Collins at NCGRP, by the way, it's a USDA facility, is the germplasm. And what germplasm is, the little bits of life. Mostly we're talking about plants. And what we want to do is collect the tiniest little bit of life that captures the genetics that you just heard from our speakers. And there are lots of different types of germplasm. My favorite is the seed, because that's my favorite. And that's actually what got me started in research to begin with. Seed is a tiny little individual bit, is a little packet of joy. But we can also study it by the pollen. We have animals now for about 10 years. We're storing animal germplasm in the form of sperm. Sometimes, like with the grapes, you don't want to actually look at the reproductive unit. You actually want that particular individual. And so we can actually store that particular individual in terms of plant cuttings in tissue culture, or we can take the buds and we can graft them. And we can do lots of different things with the germplasm. But the point is that it's just that little bit of life that we can then grow out and have a whole new plant. And so here are some examples of it, just growing in various ways. As you can imagine, making a large collection, there are about 700,000 accessions at NCGRP in bags, as Steve said. And managing a large collection, just like a collection of books or a collection of stamps, takes a certain amount of expertise. You have to know what's important, you have to know what's valuable, you have to know what's more common so that you can manage it. And so we have an awful lot of research going on at NCGRP about how to manage collections. And more importantly, because we're storing germplasm, what's really important is to keep it alive. And keep it alive so that you can grow it out and do those common gardening experiments or have that germplasm available for our breeders to work with. And so we have research ongoing on that as well. And one of the things that I'd like to point out is that research in cryobiology is we are the center of it for plants. Right here in Fort Collins, that type of research has been introduced by our facilities, and we use it all over the world. And with that, we're able to keep this germplasm alive. It's hard to know, but we're thinking a couple thousand years. I won't be around to say that I'm wrong, and you won't be around to say that I'm wrong, but we're pretty good at it. And because we're